From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on March 27th, 2025. Hello, Micronauts, and welcome once again to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 85th episode of Matters Microbial. 15 more episodes to go before number 100. Thanks as always for listening, viewing, and sharing the good microbial word. There's been a lot of interest in our Swab story experiments. One of my undergraduates, Chiana, swabbed a new water fountain in the building and found the following. As you'll remember from last time, similar appearing colonies? Most likely. And I doubt they have much to do with us or anything we do. Several sessions ago, I discussed what appeared to be a white colony from a water bottle refill station outlet that appeared to be antimicrobial. So, following the Tiny Earth protocol of Joe Handelsman and co-workers, I tested it by seeing if the white colony could inhibit other bacteria. Isolate number 1 seemed to produce a small zone of inhibition against E. coli, as seen here. When isolate number 1 was tested against Bacillus subtilis, a strong zone of inhibition resulted, as seen here. And when isolate number 1 was tested against Acinetobacter, there appeared to be no zone of inhibition, as seen here. This shows that the antibacterial effect of isolate number 1 does not impact all bacteria, but only some. I suspect it's used by the bacterium to stake out living space in a biofilm in the refill station pipes. Something to think about. It's also possible to test a number of isolates at once using the same approach. So, let's test a number of isolates against E. coli as seen here. Please remember that L stands for the lichen isolates, W for the water fountain isolates, and R for the water bottle refill station isolates. No effect seen. Oh well. Trying the same approach against Bacillus subtilis, lichen isolate number 1 produces an odd zone of inhibition, seen here. And finally, against Acinetobacter, lichen isolate number 1 seems to create a solid zone of inhibition. This is suggestive to me that perhaps the lichens are using some microbial inhabitants to exclude others to help in development of the lichen or its preservation. Sometimes nutrient media, dilutions, and similar fine-tuning approaches are necessary to see what's happening. But the most important thing, listeners and viewers, this is how students in my classes get to discover and analyze a microbe that they isolated. I can't wait to update you further. During several prior episodes of this podcast, we've explored how human intervention and changing climate can impact microbes as they relate to cities, hospitals, and the ocean itself. Nearly 1,000 years ago, Polynesian voyagers arrived in the Hawaiian Islands from various locations in the South Pacific. The indigenous culture that developed and prospered on those islands had a close and interrelated relationship to the ecology of this region. I have long been fascinated by the microbiology of islands and coral reefs and the diverse and too often threatened ecology that exists there. For example, the late Mary Ewell co-wrote a wonderful book titled Coral Reefs in Microbial Seas, linked in the show notes, that discusses how the microbial ecology of coral reefs can swiftly change due to weather or often human intervention. So, I'm beyond honored and very grateful to welcome Dr. Rosie Alegado of the University of Hawai'i, Mānoa, to the podcast to discuss her laboratory group's research into the microbiome of indigenous seascapes in Hawaii, as well as the philosophy that defines her work. Welcome to the Quality Quorum, Rosie. It's truly a privilege.
Hi. Hi, Mark. Good morning. Thank you so much for inviting me to come and
I really appreciate you with the 3-hour time difference. I really appreciate you being here. Thank you.
No problem. No problem at all.
I know, Rosie, that you wanted to say a few words about the great Dr. Isabella Abbott, and I put links in the show notes. For listeners and viewers, do you recall the Luxart I talk about so much? The bacterium that produces the light, Photobacterium lignothae KNH6, was isolated by doctors Ned Ruby and Eric Stabb from Kaneohe Bay, Oahu. So my wife, Dr. Jennifer Quinn, produced 2 Luxart portraits of Dr. Abbott in her and your honor, Rosie.
Thank you so much.
So please tell us about Dr. Abbott.
I'd love to. So Dr. Isabella Kea Iona Abbott was the first Native Hawaiian to receive a PhD. And she was such a huge influence to so many Native Hawaiian, pretty much all Native Hawaiian scholars. She grew up in Hana, which is a very remote area on the island of Maui, and learned about seaweed, and the native seaweeds, the Hawaiian word is limu. She learned about limu from her mother, who really taught her about the medicinal uses of that. And what I find so fascinating is she took that cultural knowledge and used that as a driving desire to learn more about and explore the taxonomy of seaweeds, reds, greens, and brown seaweeds, and really has wrote the definitive, many of the definitive works of that. She very early on got her PhD, and then was, for a long time, she was a research associate at Stanford's Hopkins Marine Center before she returned to Mānoa. And she was very famous in developing a course called Botany 105, Ethnobotany. And this course was a huge survey course. So she taught hundreds and hundreds of students. And the reason why I wanted to bring that up is because that course brought together, her very foundational identity, which was learning about the cultural uses of seaweeds and putting that together. Well, it was more than just seaweeds. It was seaweeds and other native plants, and really also exploring that from a scientific Western point of view as well. And so, that really introduced and I think validated a lot of the scientific knowledge that we hold in our native cultures, and really brought that forward for a lot of students here at the University of Hawai'i. So, that aspect of her as a teacher is unparalleled, but, she is just so well known as a taxonomist. She described, I would say, really, this is not an underestimate, well, at least half of our Hawaiian plant marine flora. She published 160 papers, 6 books. And so I think she very much set the standard of super high excellence for Hawaiian scholars, and really she's my superhero. She's my superhero, and really the superhero of so many Hawaiian scholars today. So thank you so much. We celebrated her 100th birthday in 2019. We had a special issue in the journal Pacific Science. And so I encourage folks to check that out, because a lot of the papers there were about legacies of her work in taxonomy in phycology. So thank you so much.
Oh, no, I find a lot of students aren't interested so much in the history of the sciences. And I really think looking at the path that was trod before helps inform how we move forward. So I want to thank you for this opportunity.
Yeah, thank you. She had so many firsts. And so that's so wonderful.
Rosie, I think it would be a great opportunity if you talked a little bit about what's behind your research and what you found. And there are going to be lots of terms that are Hawaiian based on indigenous ideas that I hope that you can explain briefly to us.
Sure thing. So I direct, instead of calling myself the PI, I say that I'm the director of the Microbial Ecology and Evolution Lab in Hawaii. And our acronym is the ME'e Lab. And the Hawaiian word me'e actually means hero. So it's kind of very similar to how, Mark, in your classes, you have your micronauts, and I always see them as little superheroes, the microbes. So we're the ME'e Lab. And what we do is that we study the microbiomes of indigenous seascapes. And you can see in this slide, what I'm showing is actually the image of a Hawaiian structure. It's called a hale, and it's overlooking a fish pond. And the Hawaiian word for fish pond is loko'i'a. And the reason we study that is that we are looking to understand the eco-evolutionary processes that are going on in this pond. So in this next slide, which is our conceptual model, what we're doing is we are trying to look at how indigenous management really influence both the genotype and phenotype, which is kind of some of the evolutionary aspects of these systems, as well as the ecosystem or the ecology of these systems. And our idea is that we bring together studying abiotic factors. Some people might call them water quality, some people might call them temperature, and the habitat structure. In other words, what is the trophic interactions and how is the trophic food web constructed? In order to really understand how human activity, which in this case is primarily indigenous guided resource management, and how that is all interacting together. And so the folks who are restoring these natural indigenous seascapes are fish pond practitioners, and their goal is to use indigenous centered or biocultural restoration to restore the original land uses. And you'll also see in this slide a couple of other Hawaiian words. One is kaia'ulu, and kaia'ulu means community. And it's also to restore a bigger image, which is the lahui, or the larger society. And there's this overarching broader concept called aina momono, which really just means abundance of our land, seas, and waters. And the reason why we think about that is we have a slightly different, some of your listeners might be familiar with the One Health concept, which kind of looks at how is the intersection between animal health, people health, and ecosystem. But we have a different concept. We really think about it in terms of ola ka'aina, ola ke kanaka. And ola means life. So when the life lives, people live. And we see that as a nested ecosystem as opposed to overlapping Venn diagrams. So at the middle of it is us, people, Kanaka, the individual. And of course, even within that, because we have nested ecosystems, is our microbiome. But of course, that microbiome, kind of pervades throughout these nested ecosystems. And so we have nested ecosystems of family, community, society, and lands and waters. And they feed forward and feed back. So we really feel that our underlying principle is that those are what kind of make us who we are. So that's kind of our microbial framework, is that we work off of the idea of nested ecosystems that are all built on a microbial foundation. And we use that in order to work in these natural ecosystems, really natural microbial observatories, they can be thought of, in order to study how our human activities on the land and in the waters are altering microbial ecosystems and altering the functioning of these ecosystems.
There's a term that's being used a lot by Seth Bordenstein and coworkers, the idea of the holobiont. And I was struck how much this reminds me everything is interactive. And that's both a philosophical idea, and it's also apparently quite true.
Yeah, absolutely. And I think some may say that's a universal thing that we continually rediscover and actually maybe even tweak and see how it reapplies to different ecosystems. And so, as you mentioned before we started talking, that I had a whole nother life that was very lab centric and in the lab and focused on, really beautiful questions. And that is still a part of my lab, to kind of think about the evolutionary origins of animals and how that is based on microbial interactions. But what this project that I've kind of really launched on in the last 10 years is to enable microbial data to inform our understanding of larger larger scale environmental change. Because when we think about, and I'm going to use the phrase climate change, microbes actually, I believe, play a really fundamental role that is not as well understood. It's a black box, because when we think about something as big as how our Earth systems are changing, microbes are literally a black box, but they play so much fundamental roles in terms of functioning and cycling of our nutrients, and just all of the layered kind of activities up above that. So our goal when I moved home was to, and really became reconnected to these spaces, was to now utilize baseline observational data to track some physiological changes that are occurring in response to the natural environment. And then not only just be out in the field, but to pair that. So my lab really pairs field experiments along with lab experiments and perform complementary experiments to understand what are the underlying processes that are influencing and at play with these interacting ecological scales, and then to develop, and this part is still more in the early stages because it takes a long time to get enough data to do that, but our long-term goal is to develop predictive quantitative modeling that brings together our lab measurements and so on and so forth. So I wanted to share today the field aspect because I think folks who have read my work might be more familiar with my lab work, but not as familiar with the work that I've been doing in the field. And the other reason why I wanted to share the more field-based aspect is because there is a lot of field work going on, and our approach is very much to work with our community and to bring them in as co-partners and co-experts in knowledge co-creation and really the way that we design the experiments. So in my next slide, I really wanted to introduce to you all the ecosystem as a whole, not just the one pond where I'm sampling, but it's really on a watershed scale. And that is the indigenous social ecological ecosystem of what I call the Ahupua'a. And it's not something that I invented. I just want to be clear. This is a very historical management unit that was used by Native Hawaiians in order to have what's called ridge to reef management. So in Hawaii, our mountains and reefs, we have the advantage actually of being really short in terms of the distance between them, and they're connected by water. And so what happens is that rain will fall down on the top of the mountains and come down through the ecosystem. And as it comes down, it's picking up nutrients, right, from the land, from the terragenous inputs, from runoff. And Native Hawaiians utilized those increased nutrient inputs in order to do a couple things. One was they used it specifically to put it into agricultural systems, in this case primarily in rain-fed ecosystems. It was taro, Colocasia esculenta, which is a very important food crop in Hawaii, but across the Pacific, across Asia, even into Africa. And so that had a really important input, not only for growing food, but also for slowing down the water, helping the sediment to come out. So it kind of cleaned the water system. It was this amazing multi-purpose thing. And the microbiology of that is fascinating. Haven't really super gotten into that. But from there were still nutrients left over, and that helped to feed nearshore ecosystems where Native Hawaiians, again, engineered estuaries, which we now know, which I'm considering to be fish ponds, in order to maximize primary productivity of phytoplankton, but also seaweed. That's connection back to Dr. Abbott. And so you have these managed, really engineered ecosystems to grow both your carbohydrates as well as your protein. And that is what enabled us to remain resilient for centuries without the help of Costco and Safeway and Target and all of those things. And so it was an enhancement of the natural food system. I don't want to kind of romanticize it and be like, we were in harmony, hold on. There were definitely, some ecosystem disruptions that happened, but they were very much limited to the coastal areas, and they were not built upon introduction necessarily of a ton of new species. It was very much learning and observing over multi-generations what were the species that were doing the best and how to enhance the productivity of those regions. And to me, that's some fabulous microbiology as well as ecology ecological knowledge that they had to really create and build upon. Yeah, and
You,
so yeah.
I was just going to say, and again, I'm sensitive to the idea. It's easy to romanticize cultures and ideas that I haven't grown up around. So I'm not trying to do that either. And I appreciate you saying that. But it does sound like more of a stewardship than I'm used to. And
Totally.
that's
Yeah, I would say it's even beyond stewardship. I would say it was a kinship relationship that we
wonderful.
really recognize that, for example, the taro plant that I referred to, the Hawaiian word is kalo, that it is like our older brother. And so you have to take care of your older brother and it will take care of you. And even in our stories, our mo'olelo, which is the Hawaiian word for stories, and our ka'au, which is our um, kind of really epic tales, um, we recognize that the kalo is our older brother. And when you have an older brother, the older brother takes care of you and you take care of it. And in this reciprocal relationship, that story tells us that it's upon us to take care of our environment so that it can continue to feed us, for sure. And there are also stories of what happens when we don't do that and we break the system. Then we get in trouble and there's famine and drought, and that happened too. But we learned our lesson and then we didn't do that again. We innovated on that and got smarter and more judicious in how we managed our ecosystems. Yeah.
Oh, I'm thinking about, I'm so interested in the microbiomes and the kinds of things that are there and
Oh,
what
for
they're doing.
sure.
Yeah.
Yes, yes. And so in my next slide, when we think about what does that fish pond food web look like, this is just a very, this is like a beautiful cartoon figure, but um, the what Hawaiians decided to do was, I think many folks today are familiar with Hawaiian coastal food systems. Maybe you've gone and had a poke bowl, right? Though that is generally made of ahi tuna, which is a apex predator. Well, we weren't trying to do that in Hawaiian fish ponds because if you know about ecosystems, if you're trying to grow an apex predator, it takes a whole lot of biomass in order to do that. And Native Hawaiians figured that out and they knew that and they realized, you know what? It doesn't make sense for us to try and grow these pelagic organisms. In fact, it's not even possible. But what we can do is we can grow these reef fishes. And in particular, they were growing, to be honest, fishes that are really important to the global food system today that I think maybe a lot of folks in the United States aren't as familiar with. They were growing mullet. They were growing Pacific threadfin. So they were growing reef fish. And the way that you do that is it's really smart and efficient on a number of ways. One, as I said, it's not an apex predator. You don't have to feed all of those things that you'd have to grow fish in order to feed fish, right? Here, they were actually, we've come to understand is that they were seaweed farmers or phytoplankton farmers. They were growing the base of the food web, so basically vegetarian fish. They were trying to maximize phytoplankton production. And of course, in order to do that, you have to have really good understanding of the nutrients. And what I find to be so astounding is that phytoplankton, if you're trying to maximize that and the nutrients, that's all microscopic. You have, unless you have a phytoplankton bloom, you're not really seeing it all the time. And so to make that connection, not only between the nutrients to the phytoplankton, but maybe also understanding that it wasn't only the green folks. Sorry, I tend to personify, anthropomorphize microbes a lot. So it wasn't only the green people or the phytoplankton, it was this understanding that you also have these heterotrophs that then
Mm-hmm.
get eaten by zooplankton, and that might also sustain these lower trophic level food fish. And so what's interesting today is that if you look at contemporary aquaculture, for a long time there was this idea we're just going to feed and grow monoculture, but they're expanding into newer technologies called integrated it's these integrated systems are trying to grow things at different trophic levels, and that's what this fish pond is. The fish pond, this indigenous mariculture system really does that. You have multi-trophic food within one system. So you have primary consumers, secondary consumers, primary producers, as well as detritivores, which are we're talking like the crabs and the shellfish, right? And the filter feeders, they're all growing in one pond. So when I say a fish pond, I don't actually just mean fish. I mean everything, and you can harvest at many different stages and food types. Yeah. And so when I moved home, I really was fascinated. Actually, it's a crazy story. I was trying to look for choanoflagellates in Hawaii, and I was thinking, what are the ecosystems where they would be most likely to be found? And so I ended up doing some historical reading around when were the first choanoflagellates identified, what kind of habitats? And I realized, hey, well, we don't quite have the marshy estuaries as much as we used to, as they do, on the continent. But we have these fish ponds, and I have friends who work at fish ponds. So that's literally what I did was I called up my friend, who then I found out was an executive director restoring this fish pond. And I said, hey, can I come over and, look and try and isolate these random things called choanoflagellates? And they said, okay, because we know you, sure. But you really should come and see what this is all about. So I went to visit this fish pond. And I have to tell you, okay, I'm going to keep it PG for your listeners. But I went there and I thought, wow, I am a real dork. Of course, in my head, it was a different word, so just you can substitute that. I am a real dork. I came here just to look at whatever organism that I wanted. I was just here to just grab my stuff and get out of here. And when I got to this place, I was like, this is so beautiful. What am I doing? And right away, I just actually put away any thought of like, today I'm going to, I thought, I got here, I'm going to catch my stuff, I'm going to go back to my lab. But I thought, you know what, let me just put my sampling stuff away. And I said, I'm so sorry. Can we walk around this fish pond, and can you tell me about this fish pond, and can you tell me how it works? And they said, we immediately started having that conversation. And so this slide on community partnerships really came from that. And these are the staff of the Native Hawaiian organization called Pai o He'eia. And for the past 2 decades, more now, they have been working on restoring an 800-year-old fish pond back to its full functioning, not only in order to feed the community actual fish, but to feed the community in terms of learning how to maintain these cultural practices, feed the community also in terms of, in these times, just building that network of a community of care, of people coming together and learning how to remove invasive species and learning how to rebuild walls and all of that good stuff. And so they, for the past 10 years, have been my community partners, my scientific partners in all of this. And I really, really feel that they have provided me with so much insight and guidance, because as a scientist who's at the main campus and teaching classes, I'm not at this fish pond every day. I can't see the changes, but they're there every single day. They have their finger on the pulse of what exactly is going on in this ecosystem. And so by having this relationship with them, I can understand what's going on when I'm not there. They can also tell me, like, this is an emerging issue that we need to begin to study. There's this weird phytoplankton bloom. It was purple. Why is it purple? What's going on? And we can, and so it has transitioned more now from a, hi, can I just come and take some samples, to, hey, maybe let's study the system and figure it out, to now I've been useful to them. And they, it's been a very rich partnership where they're like, hey, we actually want to try this out. Can you come and monitor what's going on from the beginning to the end to see what are the changes to the microbial community that are happening and to different phytoplankton species? And so altogether, what we do now is in the methodology that I've developed is, I call it Aina-oriented, meaning that it's very oriented towards the land. And the word Aina actually means more than land in Hawaiian. The word ai, the base root ai, means that which feeds. So, Aina actually could be anything. It's not just land, it's water. It could actually even be your person. Your family is your Aina because they feed you. But my overall methodology of how I changed, it really changed my point of view of how I approach science, is to be very much asking the question of what is going on with this land, this foundational thing, and it's instrument based. And so over developing this relationship with these practitioners, what I've come to realize is that even in these fish ponds where they're trying to restore, it's in recovery as well, because Hawaii has been in the global sphere from 1778 when Captain Cook came here, and then, we had plantation agriculture and then development. So there's been a lot of land use changes. And so originally when the Hawaiians came, they created these ecosystems that were really high biodiversity and really, really productive. Then over time, because of land use change, we know that it had much lower productivity, much lower biodiversity on all levels. And so we know that from speaking to our elders and getting that elder knowledge. That's called ike kupuna. We know that from Hawaiian language newspaper archives. And what we're going for is they're trying to utilize that as well as local ecological knowledge to really build back up that ecosystem, to go back to where we were and increase the productivity. And that is what Aino Momona is. It's restoring and recovering this past productivity so that we can, be more resilient in Hawaii. And so they're bringing to bear that, as well as some techniques and tips and tricks from contemporary aquaculture. Technology is good and it can be helpful. And then added to that is the stuff that I can bring, which is the measurements and the monitoring to really tweak that system. And so, when we look at what is this fish pond then, and this is in the next slide, what I really look at the fish pond as a way to look at both macro and microbial dysbiosis, right? So you can think about it. Dysbiosis is often a term that is used in the person system, right? And looking at a person's metabolism, that, leaky gut, or their diet is bad. And that's pretty much in the same way of what we can see as of the fish pond. This fish pond that I'm looking at, this is Hei'ia fish pond. This is the 800-year-old fish pond. It has inputs. So the way it works is it's not a closed system. It has a wall that helps them to control flow, but it has both freshwater inputs and saltwater inputs. And those inputs are called makaha. And the word ha means breath or essence of life. And maka means the beginning. So makaha means the beginning of breath. And what that means is that you have exchange of water into and out of the fish pond. So this is a tidally regulated system. And that's why I consider it a built estuary. And so what happens is you have tidal influences. So at different times of the day, you might have more salt water coming in, and at different times of the day, you might have more fresh water coming in, and they mix together, and you have land nutrients as well as ocean nutrients, and that's what's going to produce this really rich ecosystem. And so this ecosystem, though, sounds great, but it was a little bit broken. So there were some invasive species. You had cattle egrets that were pooping in the pond, putting in all kinds of crazy microbes. You had mangrove. Now, in many other places in the world, mangrove is great, but in Hawaii, it was introduced and has totally screwed up our ecosystem. And so it was basically a big clog or plug that was preventing the way that our ecosystems work. And then we had storms that came in and broke the wall. So you can imagine this was a leaky gut. And so that was the question, that we had. And so in the next slide, I'm showing that what Paipaio Hei'ia had done is to use bring the community in and use this as a tool for educating about invasive species, a tool for how do we rebuild these walls in a very cultural way. And, they could have brought in, they could have brought in really big instrumentation, but instead what they decided to do was to bring in people and to really make it on a people to people scale so that we could actually have that education for ourselves. And so this is actually a picture of the fish pond right behind me as well. And my next slide really shows what this recovery looked like. This is a map, and the purple squares show you the progress of the restoration over time. So it's a chronosequence starting in 2007. You can see they went right along the wall clearing. And what that looked like was both removing the invasive plants that had broken the wall. And so you can see that difference between 2009 and 2000, or maybe it's 2003 and 2011. My eyes are not that good. And then in the next slide, you can see not only did it involve the removal of invasive species, but in some cases, the actual repair. And so that's really why I kind of leveraged my thinking of from from my graduate work around how to think about this as a gut ecosystem, but on a much bigger
Mhm.
scale. So yeah, you can see what happens now when you repair a body, right? And when you are in the process of repairing something from dysbiosis, what are all the changes to the microbes? And so that's what we were wondering. But of course, over this period of time, not only were we repairing it, this is an open system. There was a lot of other things going on. There were storms that were coming in, right? There were rain events, there were flooding. We were having some really large tides, which here we refer to as king tides, but also we were seeing that we had warming events. We had El Niños happening. And so we had this opportunity. I'm just going to keep saying opportunity because, if this was in a lab, we would be able to clearly disentangle and separate each factor, but we couldn't because this is an open system. And so it's a really wonderful system that's kind of overwhelming at times with all of the different inputs. And so, you know, at any point in time from the marine side, you might have a much warmer water that's happening seasonally, but also if you have multi-annual cycles like El Nino. From the terrestrial side, we have variable runoff that happens sometimes.
Mhm.
We might have pollution that is affecting it, invasive species. And so that is all changing the biogeochemistry, which of course changes the microbes of who's there, what are they doing? And a really basic question we had was, is there even like a microbiome that's stable in this fish pond? Or is there a seasonal microbiome? That's totally fine too. So the way that we chose to study this was to, in my next slide of how we study this, it's the same slide, but now really focusing on not just the questions, but the how is taking lots of water samples. So we set up a network
Mhm.
across the fish pond and we sampled across those sites, oftentimes with our community partners, and our community partners informed us of which sites would be most useful. And what it looks like is we just get water and we do lots of filtering and lots of isolation of genetic material and sequencing. And in the early days, we really focused on the bacteria and microbial eukaryotes. So we did filter out for size, and we utilized, and maybe other folks have talked about this on your show, so I'm not sure how much in detail I should go. But as you know, we use conserved genes, ribosomal genes, the 16S gene for bacteria, and the 18S gene for eukaryotes. And that tells us who's there, when they're there, and in what relative ratios that they're there. Do you think I should go more into detail about that?
No, I think, I'm watching our time as well, but I want to say that we've talked about this kind of approach. I've used that kind of approach. I've talked about it. This is beyond wonderful. You're asking if there's a core microbiome.
Yes. Yes. We want to know if there's something that's stable there that really defines it. Our hypothesis was that there had to be because, or at least historically that there had to be. And so that was part of our question was, as restoration is occurring, are we seeing a restoration back to what it used to be? So that's exactly what we did. We decided to establish a time series. So this is also bringing together some of my work now in the oceanography department. That's what a lot of our oceanographers do. We do a time series. We look at what's going on, and our question was, at least seasonally, maybe, and maybe
Mhm.
even longer, do we see different periods of time because of different predictable changes? And then moreover, we had a second question, which is, can we begin to look at and construct these networks between the trophic levels, and particularly at the microbial base of the food web? And of course, I'm not going to say that I never stopped thinking about choanoflagellates, because I never stopped thinking about choanoflagellates. And so my hope was, can we actually see what's going on with natural populations of choanoflagellates? Because I have this really big, just going to take a 1-second side quest. I have this really big question of, okay, in the lab, we have this beautiful phenotype of multicellular development, but what does that look like in nature? Do we actually ever see multicellularity in nature, and when and why? So that was my original question of why I even showed up at the fish pond many years ago. And I hope to continue to, that is being revealed as we do these works. It's too early for me to tell you anything about that. And then the last question we had was not only about the baseline, not only about the connections, but now when we have disturbances, natural disturbances, are we seeing shifts? And that's also important because another question we have is, if we have indigenous management now, is the ecosystem more resilient because we are returning to the practices of our ancestors? So yeah, I mean, that's what something that we were doing. And in the middle of that, we were able to serendipitously capture and study a really big storm that had happened. I'm going to tell you about Tropical Storm Wali. And in this slide, what I'm showing you here is a map of the Hawaiian Islands. And one thing you should know is that in the middle of the Pacific, we get threatened by a lot of storms and hurricanes. And so it's not unusual for us to be under hurricane warning or hurricane watch. But the beauty of the way that our weather system has been working for the past couple centuries is we have this thing called the trade winds, and they usually will shear off and take care of a lot of really scary storms. And we also have these really big mountains on the east side of our island chain that also are really effective at that. And what's changing under climate, because our climate is changing, is that our trade winds are getting weaker and our oceans are getting warmer. So the storms are getting stronger and they are much more likely to hit us. And this is an example. So Tropical Storm Wali formed and then became an actual storm that was named. So it got its name. And then it kind of dissolved into a remnant. But what we're learning now, especially seeing this in Hawaii, is that the storms will they'll get deorganized. They won't have a very strong, nice eye. And I think we've tricked ourselves, just like human society, into thinking, oh, if it's decomposed, it's not really dangerous anymore. If it has its name, it's something we got to pay attention to. And the problem is this was still carrying a ton of moisture and a ton of water. And it dumped a ton of water. It was still a really bad storm. That's for other, people to kind of think about. But I do think that our practice of naming things really tricks us into thinking about when we should pay attention. And I just think we got to pay attention all the time. So this bottom panel of that slide, so you see the map of where it went and came over the Hawaiian Islands. This is a map of the rain. And there's a little star under the red thing, and that's the fish pond. And so on the next slide, because we had instrumentation in the fish pond, you can see what exactly happened. These are the physical changes that happened to the pond. We had instrumentation that was measuring current, it was measuring temperature, and a whole bunch of other stuff. And so you can see this was a thing that brought wind. It brought 6 inches of rain in 24 hours, which doesn't seem like it's a lot, but it's basically a flash flood. The stream flow also changed dramatically. And whenever you have fresh water coming into ocean water is warmer, at least in Hawaii, and the stream water is much colder, right? So that's a huge selective change onto the microbial community, really anything. It's really fresh and really cold. And the temperature took a while to recover. And what's really crazy is the salinity took a super long time to recover for microbes. It took 19 days. If you think about how fast or slow microbes are multiplying, that's a really long time. A lot of things can die. A huge sweep can happen through the population. With it, you could see that turbidity, which is how cloudy the water is, how much dirt and material are brought in, that increased a ton. So you can imagine this was something that brought a flood, brought a lot of dirt with it in that water that was like muddy water, muddy fresh water, and really changed the way that the fish pond was for a number of days. And, if you're a fish, maybe that's like not a big deal. But if you're a microbe and you're like multiplying every so many hours, you're like in shock, like it's major shock. What I'm going to do is I'm going to skip over to the slide where we're looking at what happened to two different populations. First, the bacterial plankton. And I'm using the word bacterial plankton because, again, we're only looking at the stuff floating around in the water column. There's bacteria everywhere, right? In biofilms, in particles, but we're looking at the stuff in the water column. And what we saw makes sense, which is that the 2 weeks before this data that I'm showing you here is from 5 weeks. The middle week is the storm. We did 2 weeks before and 2 weeks after. And we were actually sampling all along. We sample every week, but just for the purposes of what we're talking about. So you can see that we had a dramatic increase in the diversity of what happened. And that's because new stuff that was not in the pond, it was out in the valley, in the watershed, came into the pond. So it changed who was there dramatically. And one way that we decided to visualize this was we took a tool that was used in mouse studies where they looked at they kind of did this really beautiful color coding of what were the most prevalent microbes that were there at the beginning of a study, and then they shot the mouse with, they didn't shoot the mouse, they fed the mouse or gave the mouse antibiotics and looked at how it changed the population. So we did a similar thing. So you can see, if you're taking a look at this slide, that the diversity increases, but it's increasing because new folks, which we're coloring now in gray because we don't know who they are, we just know that they're not the original people, microbes, they're coming in. So you can see that on this third week, July 23rd, now it's all gray, and that slowly over time, it might kind of recover. And so we were interested in okay, well, who are these new people coming in? Well, it turns out that the new folks coming in, it's not surprising, but surprising. It's good for us to know these things. In the next slide, we can look at, first of all, what are the environmental parameters that it's associated with, and then who specifically. The new folks who are coming in shifted the population. If you're looking at this is what's called a non-dimensional, non-NMDS, non-multidimensional. I'm sorry, I'm forgetting what the acronyms stand for, but it's a really complicated way of looking at a population of anything. And when dots, which are samples, are close together, that means they're closely related. So here in this one, we're looking at how this disturbance introduced land-based bacteria that also happen to be disease-oriented bacteria. So you have a population, then something new happens, and they're shifting, and they're really diverse. That's why all of the microbes there are spread far apart. Those are all the samples from the 23rd. And then slowly the pond is recovering. So it's really neat because thankfully the pond is recovering, but it's not great because what's happening is new land-based, maybe even pathogens are coming in. That wouldn't necessarily be a problem, except this is a fish pond where we're trying to grow fish that we might want to eat. So the big story here is that these storms kind of decrease our resilience and our ability to feed our people. But if you have a long enough time scale of recovery, it's going to be okay. What's interesting though is, so that's the bacteria. When we look at what happened to the microbial eukaryotes, which I am actually quite interested in because that's what choanoflagellates are. Well, we were really fascinated, and we're still trying to figure out what's going on. And now we're looking at the microbial euks diversity. That wasn't significantly changed. So we're trying to figure out why is it that the bacteria changed a lot, but the microbial eukaryotes did not. So one of the ways that we measure this, and I should have mentioned this earlier, is called alpha diversity. And what you can do is, it's a measure of how many different types of organisms are there. And again, I apologize, I'm doing a very back of the hand, vague, not super exact definition of it, but it's a way of saying how many different kinds of organisms do we have here. And you can see that it didn't actually change, whereas the bacteria did change. So we're in the process of figuring out why that is, and you can see that in the colors. The people, the rainbow people who are there, the microbes, they just stayed. We didn't really get a lot of new microbial eukaryotes come into the system. And so that raises questions of, how do new microbial eukaryotes get into the system? Are they replicating at a different pace? And so even though you have these changes, they're able to recover a little bit and they're not as shifted, because of those kinds of dynamics. I don't have all of the answers to those questions, but what I can tell you is the population did change. You can see in the next slide where we're looking at the microbial eukaryote and the disturbance relative, to what's going on, they changed in the same direction. So new things did come in, but the magnitude of that change really didn't shift, and they were able to go back. Yeah, Mark, I see you making some gestures. What are your thoughts on this?
I'm in love with this because I think of the environment and microbes as being reflections of one another.
Mm-hmm.
And I'm not trying to be philosophical. Too much of that when I was an undergrad, I guess. But that might be that the fish ponds have a particular setup to them that encourages that particular assembly that you need that would be resilient against change over time.
Yeah.
Rather like the cheese caves,
Oh,
where
yeah.
you tend to, and there's a term, I'm not going to say it correctly because I'm not sophisticated at all, the terroir
Yes.
of what's in there.
Yes.
Maybe that's what you're dealing with.
I think it's really cool. And I mean, for me, this is what I've been thinking a lot, which is going to sound maybe not as exciting as I've been thinking a lot about replication rates. I'm seeing here microbes, like bacteria replicate really fast. So we're like seeing something happening really fast. Whereas microbial eukaryotes maybe replicate a little bit slower. And
Right.
so we're, maybe if we zoomed in or zoomed out, we would see that a similar processes are happening, but I don't think so. Yeah, I think it might be something like what you're talking about, is that there is this overall kind of selective environment that has been created. Anyhow, the work continues. And so we learned a lot. So I want to kind of give you a summary of what we learned. What we learned is that this Indigenous-led stewardship is really, really critical. Surprisingly, maybe not surprisingly, definitely for what I call macrobial. I know some people hate that, but that's what I call the big people, the big multicellular people. Macrobial ecosystem resilience, but also microbial ecosystem resilience. And what we saw, because we saw this change, but it comes back, I think that what we have is the beginning of evidence, and we now have much longer time series that I won't talk about today, that really kind of help to support the idea that we have a core microbiome that is sensitive to change, but can come back, right? So they're resilient to these stochastic perturbations. We saw that for some species, and I didn't go into detail, that for some groups, we did see a shift to a different new population. And so we kind of like saw all of the things, it wasn't just like, yes, we definitely saw this didn't happen. It was like, yeah, we saw some resilience. Yeah, some groups changed, and we're trying to figure out why were these groups less resilient? Why did they shift? And why did we get new things? And does that matter for the functioning of the fish pond? Or, was it just that, we replaced, one diazotroph with another diazotroph. Those are folks that like to eat nitrogen, right? So the overall functionality of the pond doesn't change, but just who is doing that might have changed. So that's also interesting to think about. And I think one of the other things that we've one of the other outcomes is that we've really learned the value of working with our community partners. We've learned the value, my students for sure, have learned the value, I would say, of humility in terms of thinking about who's a scientist, who is scientific thinking. And I think that's so important.
I mean, if you think about it, Rosie, the people who, for example, take care of cheeses, now there are microbiologists that do that, but for certainly 1,000 years, that wasn't the case. Exactly. There were people that were listening to the cheese.
Yes. Oh my gosh, for sure. That reminds me very much of a Barbara McClintock phrase that has stuck with me my whole entire life, which is getting the feeling for the organism. And that's what I say to like every single one of my students, is that sometimes we're in the middle of an experiment and they're like, what is going on? I'm like, we just got to wait it out. We got to get a feeling for the organism. I don't know. We're in the messy middle, and we'll figure it out when we get to the end of the experiment. Yeah.
Oh, I'm so adoring this idea. My mind is boggled with refugia that might be in there that help to reseed. All these things are there. What a great place for you to be. I love how you're working with the community with it. And again, I don't want to romanticize it, but you've got an oral history of looking after these fish ponds for 800 years. There's knowledge there. There's experience there. And
Yeah.
you're getting to put it, perhaps uncover what the people who did it by trial and error to develop what worked best. You're going to actually figure out what's going on.
That's true, and I think it's really amazing in that we're really building upon the knowledge of our elders, but it's for the
Yes.
future. It's really for the future. It's for us to actually return to the same level of food security that we had before. Yeah.
It's the same path. It's just different parts along it. Rosie, you've mentioned several times in this podcast some interesting Native Hawaiian terms about the relationship that they have had with the environment over many, many centuries. And do you have lessons that you could share for, I hate the term Western culture, but you know what I mean. You can just call it Mainlander. That would be fine. And more to the point, do you think that there might be aspects of this reflected in Native American culture?
Always. That's for sure. I do think, well, I don't want to overgeneralize, right? Indigenous cultures and peoples were not monolithic, but I do think that there are many principles that have connection and similarity to other places. And one of them, of course, is that we are not separate from our environment. We are a part of it. And indeed, because of that, we have a reciprocal relationship. And that means that we're both interdependent as well as have a responsibility to care for that. So that's really, really important.
It has been so inspirational to have you on this program.
Oh, thank you so much. It's been fun to be here. And, I enjoy talking with you. And even the process of re-sharing this data has made me think about new things. So I'm going to take that back to my lab today.
So what's really important for listeners and viewers to remember as we're getting done here is, did you see the enthusiasm? and that's what makes the job that I have and that Rosie has so important. I guess we're, if you'll forgive me, microbial fanatics to some degree.
Absolutely.
But it's true.
Yes.
Yeah.
Totally.
Yeah. First evolved, last extinct, right? That's the way it is.
Yes, for sure. And
So
yeah.
What were you going to say?
No, I was going to say that I really am excited about growing new microbiologists. That's all.
Yes. And there are many different paths to it. Over and over again, I hear from students, there's just one path. Look at this one.
Yeah.
And and how did that happen? It was that bolt from the blue. I'm going to sit down with my sampling. I'm going to listen to these people who've been doing this for decades, right?
Yeah.
And then their ancestors the same way, I presume. The fact is, then you sat down and actually really looked, and that's the critical part.
Absolutely. Yeah. I think it was openness on both of our parts. They wanted to know what was going on in their system. I also wanted to know what was going on in their system. And I think it is very much a combination of, sure, I've been trained on a lot of lab techniques, but the humility to know that these folks are actually there every day. They know the system better than I do. So I have to pay attention and be respectful to the knowledge that they bring. Yeah.
It's that issue of respect, something that we could all benefit from in these days.
For sure.
That's really important. And you've given a wonderful example that I hope stays with our listeners and viewers.
Thanks.
I want to thank you so much for your time again. It's been a week. And I hope to be able to talk to you in the future. I wish you and yours the very, very best.
Thank you so much. I really enjoyed this experience, and good luck. And I look forward to hearing the next 15 shows to take you to number 100. I'll be listening.
Thank you so much.
Take care.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with, as always, epic links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin. And you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Rosie Alegado is Associate Professor in the Department of Oceanography and Sea Grant College, University of Hawai'i, Mānoa, Center for Microbial Oceanography, and the Research and Education School of Ocean and Earth Science and Technology. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope you've enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.